This is the complete lecture on the blood chapter. As the blood is the first chapter in the cardiovascular system, we'll start with an overview of the entire system. The cardiovascular system, of course, includes the heart, which is the pump, the vessels, and the blood. Blood, you'll remember from A&P 1, is a specialized connective tissue with a fluid matrix. There are several general characteristics of blood. Here are three of them. The normal blood temperature is higher than the normal oral temperature. You know your oral temperature is usually about 98.6, but the blood has a slightly higher temperature, about 100.4 or 38 degrees Celsius. Part of the reason for this is that the core of the body is just warmer than the surface of the body, and part of the reason is that one of the jobs of the blood is to actually carry heat to the body's surface to get rid of that excess heat. Blood has a high viscosity. Viscosity is a measure of resistance to flow. Think about molasses pouring out of a jar. The viscosity of blood is due mostly to the red blood cells, and we measure viscosity as how much harder it is for it to flow than water. So the viscosity of blood is considered to be about 4 or 5, which means it's 4 to 5 times more viscous than water. Blood is also slightly alkaline. You'll remember that a neutral pH is 7. So the alkalinity of the blood is due to the fact that the pH is higher than 7 even when it's at the low end of its normal range. So when we talk about someone being in acidosis, technically their pH is still alkaline. So we should say they have physiological acidosis if their pH is lower than 7.35. Here's a little bit of math practice for you. We say that the volume of blood in liters is 7% of the body weight in kilograms. Well, most of us don't measure our weight in kilograms, so here's the conversion. You won't have to do this for the test, but it doesn't hurt to pull out your calculator and practice. Figure out from your own weight in pounds how many kilograms you weigh by dividing the number of pounds by 2.2. So if you weighed 154 pounds, then that would be 70 kilograms. Then to convert kilograms to liters, we're not really doing a conversion, we're doing an estimation. If we know that the blood volume in liters is 7% of your body weight in kilograms, and you weigh 70 kilograms, 7% of 70 kilograms is 4.9 kilograms, or about 5. So that means that for a typical 150-pound person, there's about 5 liters of blood. Please notice that adult males slightly higher than adult females, 4 to 5 for females, 5 to 6 for males. We're going to shift briefly and talk about the functions of blood. Most of these are pretty obvious. The blood carries things throughout the body. It transports any dissolved or suspended substance within the blood. We can also talk about the regulation of pH and ions by the blood. We will be saving a lot of the details of these functions until the appropriate chapter later on in the semester. So regulation of pH and ions will come after we study the urinary system. One of the blood-specific functions is restriction of fluid loss at injury, in other words, blood clotting. Defense against toxins and pathogens is due to the immune system, so we'll look at most of the details of that when we get to the immune chapter. However, white blood cells are such an important part of the blood that we're going to look at details of those in this chapter. And then obviously stabilizing the body temperature by dissipating heat from the body's surface. So looking briefly at those transport functions again, the respiratory gases, oxygen and carbon dioxide, are carried by the blood. Nutrients and wastes are carried not just from the digestive tract and to the urinary tract, but throughout the body from different organs. Hormones and enzymes are carried from the organ that produces them to wherever it is they're needed. Components of the immune system include not only white blood cells, but also the antibodies. Minerals, which include ions, also known as electrolytes. Buffers, which help regulate pH balance, and again, heat is transported. The transport of heat is something that your author and most books don't pay a lot of attention to, But I want you to remember that way back in A&P 1, you studied the properties of water, and one of the properties of water is that it's able to transport heat. In other words, you can add a lot of heat energy to water without raising its temperature very much. So it's the water in the plasma that carries the heat. Okay, let's take a look at what blood is made of. We say whole blood consists of plasma and formed elements. And your author gives you a normal range for each. In other words, how much of the blood is plasma and how much of the blood is formed elements. And for now, I just want to focus on the average values. 55% of the volume of blood is plasma. About 45% of the volume is formed elements. The plasma consists of water and some very important proteins that we'll be talking about, and some other solutes, as we've already mentioned. The formed elements include the red blood cells, the white blood cells, and the platelets. Platelets are cell fragments in humans, not whole cells. So if we focus in just on the plasma, more than 90% of plasma is water. And again, that water has important functions in and of itself. It carries the heat, it dissolves the solutes such as the nutrients, wastes, and ions. It suspends larger particles like plasma proteins. Plasma is one of the two major extracellular fluids. The other major extracellular fluid is interstitial fluid, also known as tissue fluid. And in fact, there's more tissue fluid in the body than there is plasma. If you have 5 liters of blood and 55% of that is plasma, then you have about 3 liters of plasma. You have about 12 liters in the body of interstitial fluid. Plasma and interstitial fluid do a pretty free exchange across capillary walls. Any water, ions, and small solutes, which include nutrients such as glucose and amino acids, as well as waste products such as urea, can easily cross the capillary wall. What doesn't cross the capillary wall is the proteins. And since there are many proteins in the plasma and hardly any in the interstitial fluid, that makes a huge difference between the composition of plasma and the composition of interstitial fluid. Let's focus in on those plasma proteins. Almost all of them are made by liver cells. They don't last very long, so the liver cells, the hepatocytes of the liver, are continually making new plasma proteins, just as a constant job. Remember that the plasma proteins cannot get out of the capillary, so there are very few proteins in the interstitial fluid. These plasma proteins have colloidal properties. Now, I'm going to send you back to Chapter 2 to read about colloids. Colloids are basically mixtures of water and particles such as proteins that are too large to dissolve, but because they have negative charges they won't settle out. These colloids that are made of proteins and water, such as plasma, affect the fluid balance of the body. We'll study that in more detail when we get to capillaries. Now, you'll notice there's one major group of plasma proteins that are not made by the liver, and that's the gamma globulins. You've probably heard of gamma globulins. That's the same thing as antibodies, and we'll study them later, but I want you to know for now that of all of the proteins found in plasma, this is a major group that is not made by the liver. So here are the 3 basic categories of plasma proteins: albumins, globulins, and fibrinogen. Albumins, you'll see, are the most abundant, 60%, and these are in fact all made by the liver. There are some differences in the different varieties of albumins, but not enough for us to worry about. Most people just refer to it as albumin and don't worry about any distinctions. The functions of albumin are pretty important. Albumin molecules have a little hydrophobic pocket that allows them to transport lipid-soluble substances such as fatty acids and thyroid hormones along with steroid hormones. Globulins we divide into 2 basic categories, and I noticed in your book that the gamma globulins are not even mentioned. And part of the reason for that is that they're in the blood for an immune function, so your author intends to talk about them later. The transport globulins are most of what you see. The transport globulins can be subdivided into different groups, and you don't need to remember the groups, but you do need to have some idea of why they're there. Obviously, they're transporting things. Some of them transport hormones. That would be steroid hormones and thyroid hormone. Some of them transport metals, such as iron. The apolipoproteins, when lipid is combined with them, are called lipoproteins. And then there are some specific steroid-binding proteins. Fibrinogen, which constitutes 4% of all of the plasma protein, is a single protein, and it has a single job, and that is to convert to fibrin, and fibrin is the stuff of clots. Now, there are proteins that are just being transported by the blood. They're not really considered plasma proteins because they don't fit into one of those categories that we just went through. So about 1% of the proteins in the plasma are just passing through. They're just on their way somewhere. And these are going to be the hormones and the enzymes that are secreted into the blood for a specific purpose. Okay, moving on to the formed elements. You have red blood cells, white blood cells, and platelets. Red blood cells, you know, are called erythrocytes. They transport the respiratory gases, oxygen and carbon dioxide. The white blood cells are called leukocytes. They're part of your immune system. And platelets are little packages that are pinched off of a cell in the marrow, and they are involved in clotting. Of all the formed elements, almost all of them are red blood cells. So that value I gave you earlier of 45% of the blood volume being formed elements, less than 1% of that is white cells and platelets combined. Almost all of that, 45%, is red blood cells. Red blood cells are packages of hemoglobin. Hemoglobin is a protein complex. It has a red pigment in it due to the iron content, and that iron will bind and transport oxygen, and the protein part of the hemoglobin will bind and carry carbon dioxide. You need to know the normal counts of red blood cells, and we're going to use the same method to count white cells and platelets, so let's pay a little bit of attention now to our units. We're counting the number of cells in 1 microliter of blood. A microliter is a millionth of a liter. And another way of looking at it is that it is a cubic millimeter. A cubic millimeter. So a little tiny cube, a millimeter on each side. So try to imagine how tiny that drop of blood is, and then imagine 4 to 6 million red blood cells packed in that little tiny drop. 4.5 to 6.3 million per microliter in males, 4.2 to 5.5 million per microliter in females. Another way to measure red blood cells is the hematocrit, and the hematocrit is also called the packed cell volume. You've probably had a hematocrit run. This is where they take a tube of blood and stick it in a centrifuge and pack the cells down. And what you see is that there's just the tiniest little coat called the buffy coat that contains the white cells and platelets floating on top of this long tube of red, which is the red blood cells, the packed red blood cells. Normal values for hematocrit in a male, 40 to 54%. For a female, 37 to 47%. Remember that is related to that formed element volume of 45%. So we'll move on and talk about the structure of red blood cells. Red blood cells, you probably already know, are little flattened discs, thin in the middle and thicker at the edge. When you look at them in a surface view from a blood smear, they look pale in the middle because they are thicker around the edge. When you look at them in a three-dimensional view, that thin spot in the middle is a little easier to see. And when you look at them in a sectional view, it's even more evident. So you've got basically a thickened ring around the outside and a flexible thin spot in the middle. And the shape of that red blood cell allows it to have certain properties that help the blood flow and to help the red blood cells actually do their job. So that shape, that flattened disc shape, increases the surface to volume ratio, which means more of the hemoglobin is close to the cell membrane of the cell and oxygen can be released more quickly. The flattened discs form stacks called rouleaux. I'll show you a picture of that in just a second. And that stack formation allows them just to line up single file and go through narrow blood vessels. And that flattened disc allows the cells, when they're young and healthy, to bend and flex when they go through tight spots in certain capillaries. So even though their diameter is almost 8 micrometers across, they can squeeze through a tight spot in a capillary that's only half their size. So here you can see some rouleau formation. There's the stacked cells, and that reduces the resistance to flow. It makes the blood easier to push through the capillaries. Red blood cells don't have any nuclei. That improves their ability to carry hemoglobin. It gives them more room, I guess, to carry more hemoglobin, but it does mean they can't do any repair if a cell is damaged. Most cells replenish their cell membrane throughout a fairly long lifespan. Red blood cells are unable to do that. So eventually the red blood cell's membrane becomes rigid and brittle, and the cell will find the end of its life, probably in one of those tight spots that I mentioned earlier. Additionally, red blood cells have no mitochondria. When you remember that mitochondria are where aerobic respiration takes place, that's the use of oxygen to produce ATP, it kind of makes sense that the cells that you have that carry oxygen to your other cells don't use oxygen themselves. So when you combine the lack of cell organelles, nucleus, ribosomes, and mitochondria, with their very simple lifestyle, all they do is zip through the blood all day, it shouldn't surprise you that their lifespan is fairly short as things go. Now, 120 days, when you think about it, if there are 30 days to a month, that's 4 months. That's longer than the lifespan of the cells on the surface of your skin. So it's really not that bad when you consider that they have no way to repair themselves. Let's focus in on the hemoglobin. Hemoglobin, you remember, is the iron-containing protein complex that transports the oxygen attached to the iron, carbon dioxide attached to some of the amino acids of the protein. We have a way of measuring just the hemoglobin. So here are the normal values for you to remember: 14 to 18 in a male, 12 to 16 in a female, and that unit is called grams per deciliter. And a deciliter is 1/10 of a liter. When we look at the structure of hemoglobin, we say that it has a complex quaternary structure. Now, I'm just sure you all remember from A&P 1 that proteins have 4 levels of structure: primary, secondary, tertiary, and quaternary. And that quaternary level is the association of different polypeptides. What we find in hemoglobin is that there are 4 separate polypeptide subunits that combine together to make a single hemoglobin. Each of those 4 subunits has a heme in the middle, and each heme contains 1 iron. The iron is responsible for binding the oxygen molecules, and we would say it binds it loosely so that you can understand that when the oxygen concentration is high, such as in the lungs, the iron will avidly grab the oxygen. But when the oxygen concentration drops, as it does in your tissues, then some of the iron molecules will give up the oxygen that they've been carrying. When you look at the structure of a hemoglobin molecule, let's get our bearings. I see 1, 2, 3, 4 subunits. 2 of these subunits are called alphas, and 2 are called betas. And we're not really worried about that at this point. However, it's important to know that when someone has a mutation to the hemoglobin, such as in sickle cell anemia, it's going to affect one of those 2, either the alpha chain or the beta chain. Each polypeptide has nestled in the center of it the heme, with the iron in the middle of the heme. Here is the heme. There's the iron in the middle. And what I want you to notice about the heme is that it is almost entirely carbon and hydrogen in a ring. And you'll remember from A&P 1 that anything that is almost entirely carbon and hydrogen is a lipid-soluble substance. So this heme is in a group of lipid substances that form rings and very often have a pigment, a color to them. So the color of heme is based on the presence of iron. If you take the iron out, it actually changes the color. Now, do you remember I told you that there were alpha and beta globins in the hemoglobin chain. In the embryo, there's actually a different variety of hemoglobin. That is, they don't use alpha and beta, they use something different, and it increases the affinity of the hemoglobin for oxygen. That is, fetal hemoglobin has a stronger attraction to oxygen than adult hemoglobin. So that in the placenta, where the mother's blood is coming close to the baby's blood, the fetus's blood, oxygen will readily leave the mother's blood and go to the fetus's blood because of the stronger attraction of the fetal hemoglobin for oxygen. So let's get some terms down. When we talk about hemoglobin function, yes, it delivers oxygen. You see we call it oxyhemoglobin. This is what I call a fake chemical symbol. HB is not a true chemical symbol. It's a shorthand notation for hemoglobin. You might wonder why we didn't call it Hg instead of Hb, and the reason is Hg already was taken. It stands for mercury. So HbO2 for hemoglobin, that is fully oxygenated. If it's not fully oxygenated, we call it deoxyhemoglobin. If it's carrying carbon dioxide, we use this as our designation, and it is called carbaminohemoglobin because the carbon dioxide attaches not to the iron but to an amino acid. And another function of hemoglobin is that it buffers hydrogen ions for us. If it has a hydrogen ion attached, we say that the hemoglobin is reduced. Okay, now you remember that the red blood cells have no nucleus, cannot do repair, and last about 120 days. So if you look at how many of them in your body are going to be at the end of their lifespan every day, it turns out it's about 1% of your total red blood cells are going to wear out every day. Now, if you sat down and figured out that you have 5 million red blood cells per cubic millimeter, per microliter, and you have 5 liters of blood, I'm sure you can do the math, that works out to 25 trillion red blood cells. So if you lose 1% of those a day, then divide that day into 24 hours and those hours into 60 minutes and those minutes into 60 seconds, you're losing between 2 and 3 million red blood cells every second of every day on average. And if you're losing that many, don't forget that you also have to be producing that many to make up for the ones that die. Still, that's a lot of hemoglobin to have to deal with. If you're losing 3 million red blood cells every second, you have to deal with the hemoglobin in those dead red blood cells. So there's a very particular process that's followed. Macrophages, which you remember are cells that like to eat things, macrophages of the liver, spleen, and bone marrow can capture old red blood cells. Remember that as they get old, their membranes become rigid. They can't squeeze through the tight spots anymore. That makes them easier to catch. And when the macrophages engulf them by phagocytosis, then they can process them and recycle or excrete the waste products from hemoglobin. If the red blood cells don't get caught and captured by macrophage, they can actually lyse. That's called hemolysis. They're going to lyse right in the bloodstream and release their hemoglobin into the plasma. So once the phagocyte catches the red blood cell and phagocytoses it, its lysosomes will combine with it. It will digest it down. The proteins just get digested down to amino acids and recycled. The heme is a problem. So we are going to pull the iron out of the heme. We're going to recycle it or store it. And then once we take the iron out of the heme, we convert what's left to a waste product called bilirubin. Remember I told you that once you take the iron out, the molecule's not red anymore. Actually, it goes through a couple of steps, but it's going to turn green. As it's excreted into the bile, bile is green in color. So bilirubin is excreted by the liver, and that means any bilirubin that was made in spleen macrophages or bone marrow macrophages have to get through the plasma to get to the liver. And bilirubin is another one of those little lipids that your albumin can carry for you. You remember one of the functions of albumin is to carry small lipid molecules? So bilirubin can be carried through the blood by albumin to the liver. Once the liver cells get all that bilirubin, they excrete it into the bile. There are little canals that run through your liver that carry bile. And so the bile then is secreted into the intestine. And in the large intestine, the colon, where you have bacteria that live, they can actually convert it into some other bile pigment-like molecules called urobilins and stercobilins that can change the color. So urobilins can actually get reabsorbed into the plasma, and your kidneys will get rid of that in the urine, and the color of those is yellow. And stercobilins in general stay in the colon and add to the color, the normal color of feces, that dark brown color. And so if you have a patient who has very pale fecal matter, then chances are they're not secreting any bile, and they may have either a blockage in a bile duct or a liver problem. Liver problems also show up because the bile pigments will build up in the blood, and as they build up in the blood, they can spill over into the skin. And so that bilirubin buildup, you remember I told you bilirubin is green, but if you have it dilute enough, as it is in the skin, it looks yellow. So jaundice is the yellowing of the skin that is associated with a buildup of bilirubin. And you see jaundice in newborns occasionally who just have an immature liver. You see it in alcoholics and other people that have had chronic liver disease, cirrhosis of the liver. Or you see it sometimes in cases where the red blood cells are being broken down too quickly. In some cases, jaundice can be so severe that even the whites of the eye will turn yellow. Okay, so you remember I said we're going to recycle or store the iron. So once the iron is removed from the heme, then it has to get into the bloodstream where you have a transport protein. That was one of our jobs of some of the plasma proteins. The one that carries iron is called transferrin, and it circulates until it gets to the liver cells that then can pick it up and store it as either ferritin or hemosiderin in the liver. And that gives you a sort of a stockpile of iron in your liver cells in case you don't get enough iron in your diet. So here we see some diagrams, a series of diagrams that's just going to show us how things are happening. Here's your macrophage. It ate the red blood cells. Some of the red blood cells died in the bloodstream, ruptured in the bloodstream, and any hemoglobin that was released into the blood is going to get picked up and removed by the urine. But in general, the red blood cells are going to be phagocytosed by your macrophages. The macrophages are going to break the hemoglobin down to amino acids, take the heme, pull the iron out, and convert it to bilirubin, and then excrete the bilirubin to the bloodstream where albumin can carry it until it gets to the main cells of the liver to be excreted into the bile. The iron was picked up by transferrin in the circulation, carried back to the bone marrow to be made back into new red blood cells. The bilirubin that's been carried through the bloodstream attached to albumin, when it gets to the liver cells, gets excreted into the bile. Bilirubin makes its way from the small intestine, not quite this quickly, to the large intestine. Urobilins can be absorbed back into the circulation, and the kidney will snag those. It will snag any hemoglobin that was released just into the circulation. So you're finally going to get rid of all those breakdown products, those pigment molecules, the hemoglobin and the bilirubin and its cousins. Okay, so if we're breaking down 3 million red blood cells every second, then we better be making about 3 million red blood cells every second. And red blood cell production is called erythropoiesis, from the Greek word poi, which is to make. Notice that's an unusual word that has an O, an I, and an E all next to each other. The tissue that produces red blood cells is called myeloid tissue. Myel means marrow, so myeloid tissue is the tissue of the marrow. And we're going to look at the steps involved in taking a stem cell to mature into a red blood cell. Our stem cells are called hemocytoblasts. And these stem cells can differentiate into one of two kinds of stem cells, myeloid or lymphoid. Now, I'm just mentioning this briefly now. We'll worry about this later, much later. The lymphoid stem cells are going to become lymphocytes. The myeloid stem cells become all the other formed elements, including not just the red blood cells, but the other kinds of white blood cells, and even the platelets are formed from the myeloid stem cells. So if we're going to make red blood cells, we're going to take that myeloid stem cell, and first of all, I want to remind you it can divide. You've got to make 3 million of these every second, so a number of these stages can go through cell division. But as the myeloid stem cell differentiates, it becomes a proerythroblast. Pro means precursor, blast means forming cell. A proerythroblast becomes an erythroblast. Erythroblasts go through 2 or 3 stages before they become reticulocytes, and the reticulocyte is the one that enters the circulation. About 1% of your circulating red blood cells are actually reticulocytes. And they're called that because you can still see just the shadow of a little bit of endoplasmic reticulum if you look at them under the microscope. And then in a few days, the reticulocytes will mature into a red blood cell. So here we see these stages. Remember, we're in the red marrow, the myeloid tissue. Proerythroblast. Erythroblast goes through several stages. At the end of that stage, it gets rid of its nucleus. It just kind of spits it out, and the nucleus will get eaten by a macrophage. And then in about a week after we started, it's ready to enter the circulation, and then a few days after that become a mature red blood cell. So this is a process that has to be very tightly regulated, and one of the aspects of regulating it is you have to make sure you eat the right nutrients. If you're making 3 million red blood cells a second, then you better have enough protein in your diet, enough iron to make that hemoglobin, and there are certain vitamins you need specifically for cell division. And they are the B vitamins, B12, B6, and folic acid. And if you are low on these vitamins, then you tend to have a type of anemia called pernicious anemia. Pernicious anemia is not common in the United States anymore because we have sufficient nutrients in our diet. But a lot of the work on vitamins was done in the 1920s, and particularly studies showing that the liver stores vitamin B12. Vitamin B12 deficiency is the main cause of pernicious anemia, and even if you get B12 in your diet, we find that in certain groups of people they can't absorb it very well for reasons we'll worry about when we get to studying the stomach. So a deficiency of B12 can't usually be solved by eating more B12 because you have stomach problems that are causing it, and so B12 can be given by injection to treat pernicious anemia. Here's another important regulatory factor to make sure you have enough red blood cells without having too many. There's actually a hormone called erythropoietin, or EPO, that is stimulated by low oxygen levels. That's a condition called hypoxia. Generally, you don't secrete a lot of EPO, but if your oxygen level gets even just a little bit low, it stimulates a very strong reaction, strong release of lots of EPO fairly quickly. And the principal organs that secrete EPO are the kidneys, with a small amount being secreted also by the liver. Okay, we're going to get into a very short discussion of blood typing. This can get confusing, so it's important to pay attention to where the different parts of the blood typing system are. The blood type of a person is determined by antigens on the surface of cells. These antigens are not found only on red blood cells. They're found on many different cells in the body, but red blood cells is where we worry about them because it's a transfusion reaction that we're usually trying to avoid, and that's why they're called blood types. They are genetically determined, and there are 2 basic groups that we look at. We look at the AB group, and we look at the RH group. Let's start with the AB group. There are 2 different antigens found in the AB group. There are 3 different genes involved. There's an A gene, there's a B gene, and there's an O gene. These are technically not different genes, but that's the terminology we'll use for now. If you have the gene for A, and only for A, then your blood type is A. Likewise, if you have only the gene for B, then your blood type is B. But because you get 2 genes for every trait, you got one from your mother and one from your father, if you had an A gene and a B gene, then you would make both the A antigen and the B antigen. They would both show up on your cells, and you would have blood type AB. If you had neither the gene for A nor for B, then that means you have 2 copies of the O gene, then your blood type is O. These antigens on red blood cells are sometimes referred to as agglutinogens. And they're called that because when antibodies combine with red blood cells with these antigens on them, it causes agglutination. Please don't confuse agglutination with clotting. We'll look at a picture in just a minute. So, this is the part where you have to keep things straight. If you have A antigen on your red blood cells, then because you tolerate your own self-antigens, and A antigen is one of yours, then you don't make A antibodies. You would only make antibodies to a blood type that wasn't your type. So if your blood type is A, then you would make antibodies against blood type B. If your blood type is B, you would make antibodies against blood type A. If your blood type is O, then both A and B are foreign antigens, so you make antibodies to both of them. And if your blood type is AB, then they both belong to you, so you don't make antibodies against either one. So as we look at this diagram out of your book, it kind of helps it make sense. Here is a blood cell with A antigen, so the blood type is A. Here's the plasma of that same person. It has B, anti-B antibodies. Blood type B has anti-A antibodies. Blood type AB, that person would make neither antibodies. And type O doesn't have any antigen on the surface, and so it produces both anti-A and anti-B antibodies. The Rh factor got its name from rhesus monkeys. It's also called the D antigen, and there's only one antigen in this group, so either you have it or you don't. So you're either Rh positive or Rh negative. And the difference between this and the AB group, the clinical difference is that you don't produce antibodies to Rh unless you've been exposed to Rh positive blood when you're Rh negative. So remember how antigens and antibodies work. If you're Rh positive, then that antigen belongs to you and you will never make antibodies against it. So the only people that have to worry about making Rh antibodies and reacting to the wrong Rh type are the people who are Rh negative. And the main group of people we worry about that in is women who are Rh negative and are carrying an Rh positive fetus. So this condition, called hemolytic disease of the newborn, occurs in cases where the father of the child is Rh positive, passes on the Rh antigen gene to the baby, the mother's Rh negative. In the placenta, the blood doesn't mix, typically, but late in pregnancy there may be some breakdown in the separation of the blood vessels, and fetal blood can get into the maternal blood supply. And the fetal blood with its antigens then can generate an immune response in the mother, and her antibodies can cross the placenta. Now, in general, you don't see a problem in a first pregnancy because it's generally at or around the time of delivery that the mother even starts making the antibodies. So typically, it would only be a second or subsequent pregnancy that would cause problems. So when you see a subsequent pregnancy, you may see the mother's antibodies crossing into the fetal blood and causing agglutination and hemolysis as a result of the immune reaction to those antibodies. So the other thing we worry about, of course, is cross reactions in transfusions. My blood type is O, so if I inadvertently got given type A or B or AB in a transfusion, then the antibodies in my plasma, remember that because I'm type O, I have both anti-A and anti-B antibodies in my plasma. So if I got given as a transfusion some type A or type B red blood cells, then my antibodies would bind to them, they would agglutinate, they would hemolyze, they would block small blood vessels, cause pain, ischemia, and potentially death. So here you see when you have the antigen-antibody reaction, it's called agglutination. The cells literally clump up, and then that marks them to be hemolyzed. So we always test for transfusion compatibility, and if we don't have blood that is the right type, then you can always give type O because it has no antigens, and type O negative is even better. So here you can see the blood testing that's done. If it agglutinates an anti-A but not anti-B, then it's A. If it agglutinates an anti-D, that's your RH. That means it's positive. If it doesn't agglutinate either one, that means it's negative. And obviously, if it agglutinates in all of them, that would be AB positive. Okay, let's move on to the white cells. White blood cells, also called leukocytes, of course, do not have hemoglobin. That's why they are colorless, and we call them white cells. They do have nuclei. The nuclei do stain with a typical red blood cell stain. And they have all the other organelles, and their lifespan can vary from just a few hours if they're called into service and die, or for decades. The job of white blood cells is to defend the body against pathogens, remove toxins and waste, and kill abnormal cells, including cancer cells. Now, there's a normal amount of white blood cells in the plasma, but in fact, most of your white blood cells have spread out. The blood is simply a way to get white blood cells where you wish them to go, and most white blood cells take up residence out in the body tissues, including connective tissue proper and the lymphatic organs, including the spleen. So what you see in the blood, 5,000 to 10,000 per microliter, is actually a very small number of the available white blood cells. And if you have an active infection, say appendicitis, then you're going to see that number jump fairly quickly. 20,000, 50,000 or more is not uncommon. So we know that white blood cells can migrate out of the bloodstream. We know that they can crawl. They crawl like little amoebas, so we call that amoeboid movement. We know that they can be attracted to chemical stimuli. That's called positive chemotaxis. So, for example, if you have an area where you had a splinter and you have some inflammation going on, the cells that were damaged actually secrete chemicals that attract white blood cells. That's positive chemotaxis. And we know that some are phagocytic. Neutrophils and eosinophils are called microphages. They eat small things. And monocytes and their mature derivatives called macrophages can eat large things. They are macrophages. So here's your list of types of white blood cells. Neutrophils, eosinophils, and basophils are in the category we call granulocytes. Monocytes and lymphocytes are in the category we call agranulocytes. And I'm sure you remember the A at the beginning of that word means without. So here you can see what they look like. Neutrophil, typical neutrophil, lobulated nucleus, a nucleus in segments. The cytoplasm is kind of a pale color with some barely visible granules in it. The granules in the cytoplasm of eosinophils stains red. The granules in the cytoplasm of basophils stains a dark blue. Monocytes, no visible granules. Lymphocytes, no visible granules. Look at the size of them. Notice lymphocytes are about the same size or slightly larger than the red cells. Monocytes, very large. Neutrophils, eosinophils, and basophils, all about the same size, slightly larger than the red blood cell. Neutrophils are also called polymorphonuclear leukocytes, or PMNs for short. Poly means many, morpho is shape, nuclear is nucleus, and they're called this because the nucleus can take so many different shapes. They are the most abundant kind of white cell in the blood, 50 to 70% of your circulating white blood cells. And the granules, which we refer to in these granulocytes, are actually little secretory vesicles full of enzymes and bactericides that when the neutrophil meets up with an area of inflammation, it releases these little packets of enzymes and bactericides and starts killing things. So neutrophils, because they're so abundant, are usually the first to attack. They release their little packets of bactericidal chemicals, they kill the bacteria, and then they start eating things. The term degranulation refers to that release of secretory vesicles. The granules show up until they release their contents, and then the granules are no longer visible. And collectively, the neutrophils and the dead and dying cells form pus. Eosinophils are 2 to 4% of your circulating white blood cells. They are very good at attacking parasites such as worm infections. So you see eosinophilia, a high level of eosinophils in people that have parasite infections, but we also paradoxically see that in people with allergies, and so someone with allergies might have an eosinophil count of 20% instead of just 2%. Basophils are the least common of the circulating white blood cells. We see that they accumulate in damaged tissue. We know that they release histamine and heparin. Histamine is important to remember because it dilates blood vessels. It's a vasodilator. Heparin is important to remember because it inhibits clot formation. Monocytes are only 2 to 8% of your circulating white cells. They're the largest of the cells. They do, as they circulate through the blood in the form called monocytes, they're not quite mature yet. Once they get into the peripheral tissues, they become macrophages, which is their final stage. Macrophages, remember, can crawl around, they can eat things, they can eat abnormal cells, they can eat bacteria, and they can communicate with other cells. They're going to attract the immune system, as we'll see when we get to that chapter. Lymphocytes, 20 to 30% of the circulating white blood cells, can move in and out of the blood. Remember that most of them are found out in your body tissues. All of your lymph organs are called that because they're full of lymphocytes. Your tonsils, all your lymph nodes, your thymus gland, and the spleen, just chock full of them, and they're scattered throughout other tissues as well. And lymphocytes have a very particular role to play in the immune system, so we'll talk about them later. There are 3 classes of lymphocytes. The first is called the T cells. They give you your cell-mediated immunity and attack foreign cells directly. The second kind is the B cells. They're responsible for what we call humoral immunity, which is your antibody formation. And the third kind is called the natural killer cells, and these are just constantly patrolling the body looking for abnormal cells, particularly those that display signs of being cancerous. Okay, so that was a quick tour through the different kinds of white cells. The differential count gives us a white blood cell profile. So when you take a blood sample from a patient and do what we call a blood smear and a differential stain, that allows us to see if they have normal levels of the 5 different kinds of circulating white blood cells, and that will allow us to determine if they have infections, if they have a high neutrophil count, for example, allergic reactions if they have a high eosinophil count. Here are 3 terms for you to remember associated with abnormal numbers of white cells. Leukopenia, anytime you see penia at the end of a word, it means deficient. Leukocytosis is an abnormally high count. Leukemia, you don't just see an extremely high white blood cell count, you tend to see abnormal cells such as the immature cells out actually into the circulation. So we'll talk very briefly about how white blood cells are produced. You remember that all blood cells originate from hemocytoblasts and that we have either a myeloid stem cell or a lymphoid stem cell. From our myeloid stem cells, we're going to get everybody except the lymphocytes. From the lymphoid stem cells, we're going to get some lymphoblasts, which are going to go out to all those lymphoid tissues and produce lymphocytes by lymphopoiesis. And again, that does not have to happen in the bone marrow. It can happen out in the lymph tissues. So in the bone marrow, we're going to see the differentiation of the other kinds of cells. The other kinds of white cells are all going to mature in the bone marrow, except the monocytes. Remember, monocytes wait until they get into the peripheral tissues to finish their last stage of development into the macrophages. We have to regulate white blood cell production. So you see here a list of various kinds of colony-stimulating factors. I'm not going to test you on the specific of each name, but I do want you to be aware of the term CSF as colony-stimulating factor as something that stimulates the production of white cells in the marrow. And here you see the diagram out of your book showing you a close-up of the whole process: hemocytoblast to myeloid stem cell or lymphoid stem cell. Lymphoid stem cell becoming lymphocytes, myeloid stem cells depending on the colony stimulating factor or EPO production, and for platelets there's another hormone called thrombopoietin, not shown here. But there you see the red blood cell production, there's platelet formation, and here are the other kinds of white blood cells being formed. Okay, so those platelets are fragments of cells in other vertebrates, such as fish, for example. They actually have whole cells with a nucleus that do the same thing that platelets do in mammals, and we call those cells thrombocytes. Thrombo refers to a clot. So sometimes we use the term thrombocyte in exchange for platelets in humans, and so you'll see that terminology associated with platelets. Technically, though, humans don't have thrombocytes. We have platelets. They're just little fragments. They don't last very long, slightly more than a week. Your spleen cells are constantly removing them. Remember, you have macrophages in your spleen, so they're just gobbling up excess platelets all the time. And you do keep some for emergencies in your marrow, so that if you have a bleeding incident, you can actually release more platelets as needed. Normal platelet count in the circulating blood, 150,000 to 500,000 per microliter. Remember, penia means low, so thrombocyte count. Thrombocyte, remember, refers to our platelets, even though they're not really thrombocytes. Thrombocytopenia, a low platelet count. Thrombocytosis, a high platelet count. And platelets are involved in various processes associated with stopping bleeding. So platelet production, thrombocytopoiesis, occurs in the marrow. The stem cells that produce them, the cells that produce them are called megakaryocytes. Mega means giant. Karyo refers to the nucleus of the cells. And so these cells are just constantly pinching off platelets into the circulation, under the control of several hormones, including one called thrombopoietin. Okay, so we've got our platelets formed. We're ready for the blood to clot. Blood clotting is called hemostasis. Please don't get that confused with homeostasis. Hemo means blood. There are three phases: the vascular phase, the platelet phase, and the coagulation phase. Now, these phases can overlap. It's not like you have to finish one before you start the next. So keep that in mind as we go through. The vascular phase is the reaction of the vessel itself to being cut. And a vessel wall has a lining of cells called endothelium, but depending on the kind of vessel, it also has smooth muscle in the wall. And if you damage the vessel wall, it makes the muscle contract, and that is called a vascular spasm. And a vascular spasm can last maybe half an hour. Hopefully by then the clot will have formed. The endothelial cells that line the vessel, as soon as you've damaged them, they actually contract, and the basement membrane underneath them has a lot of collagen fibers, and that exposure to collagen in the basement membrane triggers the next step, the platelet phase. Endothelial cells themselves actually secrete chemicals that will either promote clotting or impede clotting, depending on whether the cells have been damaged or not. So in an undamaged cell, endothelial cells secrete substances that repel platelets, but once they've been damaged, they stimulate smooth muscle contraction by secretion of endothelins, and they become sticky on their surfaces, and that stickiness helps them attract platelets, and they secrete chemical factors that will attract platelets. So here you can see the vascular spasm occurring, and the endothelial effect is going to happen at about the same time and will help promote the next phase, the platelet phase. Platelets, when platelets come in contact with either the basement membrane or those damaged endothelial cells, they can start to attach. And when they attach, they become activated. And when they become activated, they secrete chemicals and they make other platelets stick to them. And this creates a tiny little positive feedback loop where the more platelets that attach, the more platelets become attracted until you get a little plug. If you have a very small vessel that broke, the platelet plug may itself be enough to stop the bleeding. But just in case, activated platelets also release these other factors that help to stimulate clot formation. And notable here is the release of calcium ions. There's also calcium in the plasma, and as we'll see, calcium is an essential factor for promoting clotting. Well, you don't want your platelet plug to get too big, so you also have prostacyclin, which is released by healthy endothelial cells. So if the platelet plug tries to grow in an elongated way over top of healthy endothelial cells, they'll repel it, along with some other negative feedback aspects. So here you see a little platelet plug forming, remembering what started it, the endothelial cells became damaged, exposed the basement membrane, collagen factors in the wall of either the basement membrane or the outer wall of the vessel can attract platelets. So the coagulation phase begins a little bit after the platelet plug phase has started, but before the platelet plug has finished its thing. And coagulation is a separate process from platelet plug formation. The coagulation process is a cascade, and a cascade is a series of amplifying events that you can start with a really tiny amount of activation and wind up with a hugely magnified response. Let's take a look. Many of our clotting factors, most of them, are called procoagulants because they contribute to clotting. We also have another group of clotting factors called anticoagulants, which we'll use at the end of this process. There are three parts to the pathway. The extrinsic pathway is going to occur in the tissue where the blood has bled out of the vessel. The intrinsic pathway is going to occur inside of the vessel, and the common pathway occurs in both places. So you end up with this common pathway finishing the formation of a clot inside the vessel and a clot around the broken vessel in the surrounding tissue spaces. So the extrinsic pathway, because it's outside the bloodstream, has to have something that activates it, and what activates it is the damage to the cells around the vessel, and they secrete a chemical called tissue factor that actually activates the formation of factor 10. Factor 10 is one of the procoagulants. The intrinsic pathway is occurring inside of the vessel, and please notice that it's activated by exposure to collagen. Remember, that was either in the basement membrane or in the loose connective tissue around the vessel wall. We're going to have some encouragement from plasma, and notice we're still going to result in activating factor 10. So factor 10 is the first step in the common pathway. And the common pathway, starting with factor 10, you activate the formation of an enzyme which activates another enzyme, that enzyme being thrombin, and thrombin is the enzyme that activates fibrin formation, and fibrin forms the clot. So let's look at a picture that shows us more or less what these processes are, even if it's not showing us where they occur. Here is tissue damage. This really ought to be shown on the outside of the vessel. Tissue factor, clotting factor, notice the inclusion of calcium, but the tissue factor is what starts it. Add some calcium, add another clotting factor, and you get activated factor 10. Inside of the vessel, you've got some other enzymes. Notice you still need calcium, you need some platelet factors, you need some other clotting factors, and you've got some more factor 10. Once you have factor 10 activated, the prothrombinase converts prothrombin to thrombin. Thrombin converts fibrin to fibrinogen. Now you're probably wondering how much of that you need to remember for the test. And what I'm going to tell you is go look at your study tools, and that will tell you which things you need to remember. But another thing I want you to notice is that there are more steps in the intrinsic pathway than there are in the extrinsic pathway. And you remember what I said about it being a cascade. The more steps there are, the more magnified the final response will be. So you get a stronger, bigger, faster clot forming in the vessel than you do around the vessel, and that should make sense. Okay, so we don't want it to get too big, but we need to make sure that the clot forms quickly and well. So we're going to see a positive feedback loop to accelerate clotting, but then we're going to have the formation of anticoagulants, the activation of some anticoagulants to keep the clot from growing too large or the clotting process from going on too long. So here's a short list of some different anticoagulants that are actually produced in the body. There are also other chemicals that are not produced in the body that we use as anticoagulants. Most notable among those is aspirin. Aspirin is an anticoagulant. That's important for you to remember. We use aspirin for pain control, but if you take too many aspirin, you can actually get bruises because you're inhibiting clot formation. And people do take aspirin to prevent the likelihood, or to reduce the likelihood of a heart attack, and they're taking it as a clot inhibitor. Here are some other things that you need to know about chemicals associated with blood clotting. You absolutely positively have to have calcium ions for blood to clot, and you need vitamin K. Now, we haven't really talked about vitamin K before. Vitamin K is a vitamin that is required by liver cells to make the clotting factors. Not all of the clotting factors, but enough of them that if you have a vitamin K deficiency, then your liver cells can't make as many clotting factors and your blood is less likely to clot. Now, some of the anticoagulant drugs that we give people actually inhibit vitamin K. Coumadin is one that comes to mind. So if you have someone you're trying to inhibit clot formation in, you could give them Coumadin. It will inhibit vitamin K. The problem is vitamin K is found in the diet as well. So if you eat too much broccoli or other cabbage-related plants, then you're going to get enough vitamin K that you have to have your Coumadin dose adjusted. Okay, so the clot is going to retract. That's going to pull the torn edges of the vessel closer together. It's going to help stabilize the clot. And then after it's repaired, then the clot is going to get broken down by a process called fibrinolysis. And in this, there is an enzyme called plasmin that actually breaks down the fibrin. Now, it got caught up when the clot was formed, but it's a pretty slow enzyme. So it'll take it 2 or 3 days before it can dissolve enough of the clot, and by then the repair process is well underway. Plasmin is one of those enzymes that we said there are enzymes in the blood. They're less than 1% of all your plasma proteins. It's actually produced by liver cells in an inactive form called plasminogen. And we've seen that before. We saw it in fibrinogen, and we'll see it again. Anytime you see O-G-E-N at the end of the word, it means it's going to generate. It is the precursor of. And so what we have is another enzyme, TPA, that is actually produced by the endothelial cells that activates the plasminogen as it gets bound up in the clot, and so you have the ability to dissolve the clot built into the clot. We use TPA medicinally. We use TPA as a clot-busting drug. Okay, so that's it for the blood chapter. I know this was a long lecture. I hope you listened to it in segments. For the next 2 chapters, you need to use your Interactive Physiology CD. I strongly recommend you watch the CD before you try to read the book. Understanding the heart and the blood vessels is really challenging, so be sure you spend enough time on it, and let me know if you have any questions. Talk to you later.